The next topic in acute inflammation that we'll be starting with are the inflammatory mediators. Now, when we talk about mediators, it's obviously a very dry topic because it's got a lot of cramming up that you guys have to do, but we'll try to simplify this as much as possible. So when I begin with the mediators, the first and the most important thing is regarding the classification that you all have to know. So please remember, mediators are classified into two broad categories. The ones which are already there in your body from where? That is the cell-mediated mediators. Means the ones, or in other words, in order to avoid any confusion, we can call them cell-derived mediators. They are coming from some or the other cell. On the other hand, the other category becomes plasma-derived. They have come from some or the other, some organ, but they are roaming around in the plasma. So if they ask you, okay, they're roaming around in the plasma, but what is the source? The source is going to be the liver, and I'll be talking more about it. And please remember, these plasma-derived are going to have all the K-sounding things, okay? What are the K-sounding things? So please remember, the complement system is going to be a part of this. The complement pathway or the complement system, the clotting or the coagulation system, whatever you may want to call it, the clotting or the coagulation system is also a part of it, and lastly the kinin system is a part of it, or the kinin pathway is a part of it. So like I said, all the K sounding things, right? Let's repeat, we have the complement system, the clotting system and the kinin system. The Ks are a part of it. Moving back to the cell-derived, under cell-derived I have two things. One are going to be the preformed mediators, the ones which are going to be all the time present in the body, and the other ones are the ones that you will make when you have inflammation, you make them. That is going to be the newly synthesized variety. So repeating, there is a preformed variety and there's a newly synthesized variety. So there's no point in giving you lengthy lists right now. You will not register anything. Let us go one by one, okay? So under preformed, there are three things that I'm going to start off with, and these include histamine, the hero. Then we have its supporting actor, which is serotonin, and we have another thing called lysosomal enzymes. Now I want all of you to be very clear on one thing, that preformed, see, I'm telling you again, what am I talking about? Cell-derived means they are going to come from some or the other cell. Next, preformed means they are always present in your body. They are 24/7 present in your body. And these are the three things that we'll be discussing. So let's start off. Let's start off my story with histamine and serotonin. Before I start, remember histamine was given by a very famous scientist called Thomas Lewis. That is a scientist, just in case you're interested in historical aspects. But I have always dealt with histamine and serotonin as a difference table, because there are so many things which are different and then there is something which is so similar about it. So let's study them together. Firstly, tell me one thing. They are preformed, yes, we know, but where are they coming from? They are coming from some cell, remember? So can I ask you, what is the source of histamine? The source of histamine is going to be the mast cells, the basophils. Mast cells and basophils are quite similar, and also the platelets. On the other hand, if I ask you where does serotonin come from, it comes from the EC cells. What are EC cells? Enterochromaffin cells. It comes from the enterochromaffin cells and also the platelets. So I realized that the platelets are quite useful. Platelets are giving me both histamine and serotonin, but in the exam they started asking you questions of the most important ones. When I talk about the most important ones, please remember, mast cell is going to be, so basically they'll ask you what is the richest source. So for histamine, if you are asked what is the richest source, it is going to be the mast cell. And for serotonin, if you are asked what is the richest source, there's quite a controversy around it, but all said and done, enterochromaffin cell. So yes, if you are asked what is the richest source for either of these, you should be ready with your answer. For histamine, it's going to be the mast cells, and for serotonin, it's the enterochromaffin cell. Well, having said that, guys, please remember there's one correlated question for mast cell that I'm telling you. They ask you in the exam, what is a special stain for mast cells? And not only for mast cells, let me tell you, the same stain that I'm talking about is not only going to be for mast cells, it is also going to be for basophils. It is also going to be for basophils, and that is the stain in front of you, which is known as toluidine blue. The stain is referred to as toluidine blue. But now there's a twist in the story, which I have told you very categorically and in a lot of depth, even when we have been studying about microbiology lectures, that toluidine blue is a stain which is metachromatic. And I have always in microbiology lectures also given you a story, a very interesting story. What is a metachromatic stain? Forget this fancy word. It is like that topper of your class who has not studied anything at all and is going to flunk and fail and all of that. And then when he goes in the Viva exam, he's giving all answers and taking so many continuation sheets and topping the exam, right? Toluidine blue is like that, has some different color and is going to show some different color. So toluidine blue is the two-faced topper of your class, which means toluidine blue is having a different color, blue color. But ultimately, did you see what color did it give to the basophils and the mast cells? Has it given a blue color to the basophils and the mast cells? No. If you can look carefully, it's given a purplish color. So I gather from the end of it is that the basophils and the mast cells are going to get a purple color. So see, like I told you, it has a blue color, but it gives a purple color, right? That is what you need to know about these toluidine blue. It is a metachromatic stain. Coming back, yes. So we've got the richest sources. I've also told you the individual the richest source and the stains and everything. Now we talk about what is the precursor, where does it actually come from? So please, I think this you guys already know, so there's not much for me to elaborate on it. Histamine is going to come from histidine, and serotonins. What is the other name for serotonin? Serotonin is also known as 5-HT, right? So this is going to come from tryptophan. This is going to come from tryptophan. So that's the precursor. Histamine from histidine, serotonin from tryptophan. So they are so different, but where are they going to club? They are going to combine in terms of their function. So please remember, the function of histamine and serotonin is the same, and I think you already know the function. Do you remember? I had drawn some diagrams. I told you that the blood vessel is going to get dilated. So number one, it is going to help in vasodilation. Then I also told you that everything is going to move out of the blood vessels because of histamine and serotonin. So increased vascular permeability was also because of them, right? And finally, guys, please remember, is there a role in asthma also that they play? Of course, very important roles. How would someone cause asthma? By bronchoconstriction. So now please note, what are the three main events? VD, vasodilation, VP, that is vascular permeability increase, and because it causes asthma, that is why it is associated with bronchoconstriction. Having said that, this is your story of what? If I go back, I have taught you histamine, I have taught you serotonin. Their stories are done. I'm left with one more mediator in this category, that is the lysosomal enzymes. So let's start talking about them. I know it's not at all an easy table, and I take it as a challenge that I'm going to make it easy. So I'm right now very confident. I hope that confidence remains. First and foremost, lysosomal enzymes is undoubtedly a tough table. Let's make some mnemonics and learn it. You know where these enzymes are coming from? These enzymes are coming from the granules in the neutrophils. They are coming from granules in neutrophils. So actually what you guys have to know is how many granules are there in the neutrophils. I will write it as primary granules, secondary granules, tertiary granules. Very simple. Primary, secondary, tertiary, but that's not how simple it is. Examiner asks you other names. Primary granules are known as azurophilic granules. Secondary granules are referred to as specific granules, and tertiary ones are referred to as C particles. Tertiary ones are referred to as C particles. How will I learn this information? Really simple. What is the first alphabet? A. So what is the name of the primary granule? First alphabet A. So that is azurophilic granule. Easy. What is the third alphabet? A, B, C. What is the third alphabet? C. So what is the name of the third particle? That is the C particle. And finally, what do we have as secondary? So remember, S for secondary and S for secondary, S for specific. That is how I've learned it. So simple. Now life becomes a lot easier, right? So what do we have? We have number one, primary. So primary, first alphabet A, tertiary, third alphabet C, and S for secondary, S for specific. The namings are done. Now you just have to know which lysosomal enzyme is going to come out from where. Very simple again. The way you write a C particle, I want from that C, you should make a G out of it. You should make a G out of it. So what is going to come out of C particle? Gelatinase is going to come out of a C particle. That is one thing. The next thing that you have to know, what is going to come out from the secondary? See, secondary had the word specific in it. So something specific is going to come out. There is a specific type 4 collagenase. Not just any kind of collagenase. Specifically, what will break type 4? Specific type 4 collagenase and also lactoferrin. How have I learnt this information is that please remember specific collagenase hai, so specific has to come from specific, and lacto, instead of from today onwards, you can learn it as lactoferrin. Lactoferrin will always remind you that it's coming from which granules? It's coming from the secondary granules. So lactoferrin. Sorted. Now I'm just left with the primary. And what do you learn in primary school? In primary school we learn A, B, C, D, E. So I'm going to take you all the way back and make you learn A, B, C, D, E in life once again. So what is A, B, C, D, E? A stands for acid hydrolase. A stands for acid hydrolase. What does B stand for? Something that is going to help in killing bacteria. Think. Bacteria killing. What was helping, guys? Which was that pathway for phagocytosis we studied? Bacteria killing pathway. H2O2, MPO halide. Do you all remember that? H2O2, hydrogen peroxide, free radical. I know how it's formed. Chloride is there everywhere in my body. MPO. Where does myeloperoxidase come from? And there it is. The B for bacteria killing myeloperoxidase comes from the primary azurophilic granules. Very famous question. Okay, talking about the next. C for collagenase. Now in the exam, be very cautious. Which collagenase has been asked? Just a general kind of a non-specific kind of a collagenase, put it in one, primary. But if a specific type of collagenase is asked, you'll have to put it in specific granules. Other than that, remember even cathepsin with a C is going to come over here. D is going to stand for defensins and E is going to stand for elastase, and that finishes off your entire table of lysosomal enzymes. I'm not saying it's the easiest table, I'm just saying that we've tried really hard to make it easy for you. So having said that, let's do a recap. First, you have to know the names. Enzymes are coming from granules. Granules are primary, secondary, tertiary. Primary, first alphabet, so azurophilic. Secondary, SS specific, and tertiary are going to be the C particles. Okay, from the C you make a G, so it releases gelatinase. From specific, something specific will come out, so specific type 4 collagenase, and from secondary, lactoferrin will come out. And from primary school has A, B, C, D, E. So acid hydrolase, bacteria killing MPO, we have cathepsin and collagenases, defensin and elastase. But I have been a little bit smart over here, and I have actually, you will think that the only lysosomal enzyme which you guys probably knew in detail was lysozyme. And that I have not written anywhere. Where will or who will release the lysozyme? So remember, the primary and the secondary granules together release lysozyme. That is something extra that you have to learn. It comes from primary and secondary granules. So don't get confused. If they ask you primary and secondary, you will mark. If they ask you lysozyme, you will mark primary, secondary. But if they ask you lactoferrin, where does that come from? Lactoferrin. That comes from the secondary granules. I hope everyone is okay with the lysosomal enzymes, because with that you have finished. I hope by now you don't feel mediators are something to run away from. I've kind of tried to simplify it for you. Fingers crossed, I hope I've managed. So yes, they are coming from some cells. Yes, they're always present in the body. We've done histamine, serotonin and lysosomal enzymes. Now I'm going to come on to the other category. Newly synthesized means if today someone has suffered from inflammation, from today onwards it is going to form. So what are those things? Quickly making a list out of it. We have nitric oxide, platelet activating factors, we have all the chemokines, we have all the cytokines, and we have the arachidonic acid metabolites. So yes, there is a long journey left ahead of us, but don't worry, since we've simplified what we've done till now, we will simplify it further also. Keep in mind, these are all newly synthesized. Now they are going to be made in our system. So having said that, let's begin with the first one, and that is nitric oxide. And I think you guys know pretty much a lot about nitric oxide because you've read it in biochem, in medicine, and here in path. So first and foremost, I would want you to tell me or quickly revise with me the formation of nitric oxide. After that, we'll study the function. So for the formation, I think all of you know that there is a very famous amino acid called L-arginine. And L-arginine is now going to be acted upon by what? It is acted upon by an enzyme called nitric oxide synthase and is going to form nitric oxide and L-citrulline. So repeating, NOS is the main enzyme over here. Let's write it down. It is nitric oxide synthase. Before I move forward, can you tell me any other mechanism in this chapter only where the usage of arginine was very, very essential? So one arginine is needed for the production of nitric oxide. One arginine was needed in some kind of a killing process. What was that arginine, guys? That arginine was NETs. Remember? Yeah. Okay. Having said that, coming all the way back, when I talk about nitric oxide synthase, NOS, remember this NOS is of 3 types. This NOS is of 3 types, which is some extra information that I want to give you. There is something called eNOS. E stands for the one which is present in the endothelium, that is endothelial NOS. There is something called nNOS because this is the nitric oxide synthase that is present in the neurons, so neuronal NOS. And then there is something called INOS, that is inducible NOS, which you have induced. When have you induced? I learn it as I for inflammation. I have induced it whenever I need to carry out inflammation. So right now, when I'm talking about the chapter of inflammation, which NOS am I using? Am I using the endothelial, the neuronal, or the I? The INOS, the inducible NOS is what I've used. I hope you all have understood. Okay, so now I know that, okay, it's INOS, and what I have formed ahead of me is nitric oxide. Now next question is, what is the function of nitric oxide? That is exactly what they are going to ask you. So some parts of nitric oxide you know from path and some you know from medicine. So first and foremost, what is it going to do to the blood vessels? It's going to open up your blood vessels. It's going to cause vasodilatation, point number 1. Number 2, have I just now told you that can it in some form kill the bacteria? Do you remember phagocytosis? Nitric oxide was also there in killing the bacteria. So can it help in microbial killing? Yes, it can help in microbial killing. And finally, somewhere that it helps you in medicine, it is an inhibitor of aggregation, or it is an anti-aggregatory molecule of platelets. It is against aggregation of platelets. That is something that you have to know as well. So repeating, number 1, it dilates your blood vessels. It is a vasodilator. Number 2, it is going to cause microbial killing. I know that pathway, the phagocytosis, and then it prevents aggregation of platelets. That is all about nitric oxide. Quick take home message, you should know the amino acid, that is arginine. The type of NOS in inflammation chapter is INOS, and the functions. The first one is done. What is the second mediator in this newly synthesized category that you have? And that is going to be PAF. So first you need to know what exactly is PAF. It is a platelet activating factor. It is a platelet activating factor. And now remember, in the beginning I told you that hero will always be histamine. Hero is the one who's on the up front and is showing the face and is the main star of the movie. But there will always be someone who is more hardworking but is very, very grounded and doesn't come in the limelight. PAF is that. Actually the textbooks say that it is 1,000 times more potent than histamine. It is 1,000 times more potent than histamine. It is actually going to be doing much more work, but it doesn't come in the limelight. It just does its work and goes back home. That is what is PAF. So histamine is more about show off. The real work is being done by others. What is platelet activating factor doing? Everything is going to get constricted. That is how I have learned it. So when you ask me what is happening to the blood vessels are going to undergo a vasoconstriction. Remember, the blood vessels undergo vasoconstriction. Next, if you ask me what is going to happen to the platelets, if you ask me, so platelets are also going to clump together. This means there is going to be platelet aggregation. There is going to be platelet aggregation. Next, if you ask me what is going to happen to the lungs, again, constrict. Everything is going to get smaller. So there's going to be bronchoconstriction. So I all in all realize that everything is going to become small, right? However, there's a trick involved over here. Please remember, at low concentrations, at very low concentrations, PAF can also dilate the blood vessels. It can cause vasodilation. So this is a side, but a question which I have seen in a lot of Q banks. So I wanted to share this with you, that for all practical purposes, PAF causes everything less. Vessel will constrict, lungs will constrict, platelets will aggregate, everything will come smaller and closer. But when it comes at low concentrations, it can cause vasodilation also. That is what is about PAF. So nitric oxide done, PAF done. What do we move on to next? Next we move on to the chemokines. So chemo means there are some chemicals. Chemo means some chemicals. And chemokines means, is this sounding like chemotactic agents? Are they going to attract something? Yes. Chemokines are chemoattractants that you have to know. So first let me tell you what is the basic about a chemokine. All throughout a chemokine, I am going to talk about something to do with C and something to do with X. So first you should know that the C is going to refer to cysteine, okay, and X is going to refer to any other amino acid on this planet which you don't have to worry about, any amino acid. So C is going to be cysteine, X is going to be amino acid. That is sorted. Aage padhte hain. So how many types of chemokines do we have? We have something called CXC. Remove the X, what are you left with? CC. And then remove a C, what are you left with? Single C. Simple to learn. CXC. Then you remove this X from here, you remove this X from here, you're left with CC. You remove a C, you're left with single C. Remember, these are known as alpha chemokine, beta chemokine, gamma chemokine. Alpha, beta, gamma. Apart from this, there is a random weird kind of a chemokine called, the most different looking I've kept on the side. The most different looking is CX3C. So it is how many, you can count how many cysteines. It has 2 cysteines. And how many amino acids? 3 amino acids. It's the most weird, so I've kept it on one side. So first you've understood their basic terminology. Now I'll tell you their examples and what exactly are they attracting. They are going to attract some cells. So let's write it down once again. Now you know the C and the X. Are you ready to write it down once again? So first we'll start with CXC. You remove the X, what do you get? CC. You remove the C, what do you get? C chemokine. What are they also known as? Alpha chemokine, beta chemokine, gamma chemokine. Is there any weird one also? Yes. CX3C is another chemokine which is quite weird. Okay, so I get to know that. What else, guys? Now, you need to know their examples. So first I'll tell you, CXC, the example is interleukin 8. And please remember, interleukin 8 is going to attract all the neutrophils. What does it attract? All the neutrophils. How do I learn this? Because I'll tell you so many examples, now you'll get confused which one falls under which category. So remember, the way you write CXC, that's your mnemonic. The way you write CXC, you are going to make an 8 out of the X. Did you make an 8 out of the X? Did you get exactly what I'm trying to tell you? Which interleukin is going to come under the CXC category? That is interleukin 8. Easy to learn? Okay, then let's go to the last one first. Let's go to what is with C. So single C, the way you write single C, I'm going to make out of that single C, I'm going to make an L out of it. C and L look the same. Sometimes if I write C like this or if I write L like this, it looks the same. So yes, please remember, this is going to have something known as lymphotactin. The example over here is lymphotactin. No points for guessing which cell is it going to attract. I mean, the most common sense thing that you can put over here, it is going to attract the lymphocytes. So what is your mnemonic? That the way you write a C is the same way you write an L. So C is going to have lymphotactin for lymphocytes. Now, you have something called CC. Now, what is CC? Copy to. You usually, when you send an email to someone, don't you send one main person to whom you send the email, and then you have all the miscellaneous people CC, may you add those miscellaneous people, right? So what do you need to know? Now, everything other than neutrophils and other than lymphocytes that you want to attract, and not only lymphocytes will come again. So I'll give you some examples. So there are some examples over here which include eotaxin. No points for guessing that what is eotaxin going to attract. Of course, it will attract the eosinophils. Eotaxin means it will attract the eosinophils. Next, there's something called rantes. What will rantes attract? You will pick up the T out of this. You will pick up the T out of it. So this is going to attract the T cells. Then there is something called MCP, also something called MIP. Whether they say MCP or they say MIP, basically M should over here tell you monocytes or macrophages. So yes, this is going to attract the macrophages for all of us. So the name is telling me eotaxin for eosinophils, rantes for T cells, MCP or MIP is going to be for macrophages. So that is sorted. Basically all the miscellaneous things I had to put under CC. Finally, coming to the last one, which is the weird one. So it has the most weird name also under it, you will eventually realize. CX3C, I'll name a molecule you've never heard of and you will never hear again. And that is fract alkene. The name of the molecule is fract alkene. So have you ever heard of it? No. And many people call it fract alkaline. No, it is fract alkene if you read the books carefully. And what is it going to attract? It can attract anything. I'm not much interested in. It can attract T cells also. It can attract the monocytes also. So it's pretty much a non-specific kind of thing. Everything weird goes into this category. CX3C is a weird structure. It has a weird example that is fract alkene. And that is what is sorted. Can you do a quick revision for me, guys? A quick revision on this slide, a blank slide. What is the example of CXC? With the X, what did you make? You made an 8 out of it. So interleukin 8 for neutrophils. What is the single C? You made an L out of it. So lymphotactin for lymphocytes. Everything else miscellaneous into that CC email of ours, eotaxin, rantes, MCP, everything comes here. And the most weird one, fract alkene, that is going to go there. Now, all said and done, this was helping in inflammation. Understood. Is there any correlation with microbiology that you can do? Have you heard of something called CXCR4 and CCR5? Sounding pretty similar. Means CXC I have read and this CC also I have read, but why have they added an R in front of it? What is this R added everywhere in front of it? Means these are chemokine receptors. R means these are where the chemokines act. These are chemokine receptors. Now tell me, why did I say microbiology? Because CCR4 and CCR5 and CXCR4, they help in the co-binding of some virus. These chemokine receptors are the co-receptors for human immunodeficiency virus. When you study the HIV pathogenesis in micro that I have taught you, I have mentioned that for HIV to enter into our body, we need the help of these two molecules, the CXCR4 and the CCR5. Now you exactly know what is CXC and what is CC. They are chemokine receptors. Chemokines are sorted, guys. What do we have to move on to next? So what all have we done? In newly synthesized, if I focus, I've done nitric oxide, platelet activating factor. We've done the chemokines. Now we come to the cytokines. Now first, let me tell you, cyto means some of the other cells are going to give them, and they are chota, small proteins. So let me first divide it. See, when we say cytokines, there are three broad categories into which you are going to divide it out. The first category is everyone's favorite IL, means interleukins. The second category is going to be TNF alpha, and the third category is IFN. IFN is interferons. These are the three main categories that you have for cytokines: interleukins, TNF alpha and interferons. Let me finish off TNF alpha here because that's the shortest. Remember, TNF alpha should remind you of NA, no appetite. It is the one that decreases appetite in a patient. Have you so many times seen, I'll tell you, whenever we get fever, do you feel hungry when you have fever? Don't you always complain, I'm not feeling hungry, I don't feel like eating anything. As your fever goes up, you stop feeling hungry. Who and because of whom do you stop feeling hungry? Because of TNF alpha, you stop feeling hungry. Point number one. You must have seen cancer patients are very, very thin, very cachexia, cancer cachexia, right? Why are they so thin? Because they have too much of TNF alpha. They don't feel hungry. Similarly, in medicine, you must have studied septic shock. What is the mediator that helps in shock? That is again going to be TNF alpha. So please remember, a decrease in appetite, or this is also indirectly I can call it cancer cachexia. They also don't feel hungry, and this also has a role in shock pathogenesis. That is the role of TNF alpha. So before we move on with the rest of the cytokines, I believe there's a very common source of confusion that I have seen amongst the students, and that is the differentiation between, let me write it down. There is something called TNF alpha, then there is something called TGF alpha, and then there is something called TGF beta. And as many times I've seen students understand this, I see they start jumbling it all up. So let's do some basic mnemonics and finish it off. When I say N and A, this should remind you of N and A for no appetite, okay? N and A is going to be no appetite. So whenever we stop feeling hungry, that is because of NA, TNF alpha. When they talk about GA, GA should remind you of GA for gastric. This is associated with some gastric disease. Can you think of, in medicine and surgery, which disease have you heard in which the TGF alpha goes up? Which gastric disease? I'll give you a hint. The disease in which the stomach, the gastric, looks like the brain. What disease is that? Menetrier's disease. So remember, in Menetrier's disease, the Menetrier's disease is a disease of the gastric, is a disease of the stomach. So TGF alpha goes up. And when I talk about TGF beta, please remember, in GNF, I would rather learn it as this. Bahut fibrosis. Bahut fibrosis means too much fibrosis. So this is going to be the most fibrogenic cytokine that we have. This is the most fibrogenic. So I've learned it as bahut fibrosis. Let's do a repeat. N and A. When we talk about N and A, so that is what we were talking about here also. We were talking about N and A over here, right? So when I go back, when I talk about N and A, you have to talk about no appetite. When I talk about G and A, that is gastric, Menetrier's disease. And when we say TGF beta, that is going to be bahut fibrosis. Okay, having said that, now coming all the way back, we are coming back to TNF alpha. That is done. All the relations are done. Now I'm going to go on and study about the different types of interleukins, which I know all of you are very, very worried about most of the times. I'll try to simplify it today. So first and foremost, when I talk about the interleukins, I'm going to teach you in the form of a table and a question answer so that life becomes easier. So yes, can you tell me which are the interleukins that cause fever? It is interleukin 1, interleukin 6, and even TNF alpha causes fever. But along with fever, what is it going to cause that I just taught you? Along with the fact that it causes fever, it is also going to decrease the appetite in a person, right? So yes, remember that fever is 1, 6 and TNF alpha, but out of these, I hope you guys all remember this basic point that interleukin 1 is the most important, right? The most important cytokine for fever is interleukin 1. Okay, now I'm going to ask you the next thing, which is the interleukin which can act as both. When you want, it can start inflammation for you. When you want, it can stop inflammation for you. So remember, interleukin 4 and interleukin 6. 4 and 6 are both. When you want, it will start. When you want, it will stop. But the real question comes is, which is the interleukin which is only going to be anti-inflammatory, or which mediators are anti-inflammatory? Let me tell you, interleukin, there is only one interleukin. Only and only it will stop inflammation. It can never start inflammation. Anti-inflammatory is interleukin 10. And other than that, also TGF beta. Where else did I teach you TGF beta right now? That is what I taught you. TGF beta stops the inflammation. That is the reason that TGF beta causes so much of fibrosis, bahut fibrosis, most fibrogenic, because it decreases inflammation. So how have I learnt it? It's very interesting. Instead of learning it as anti-inflammatory, can you all from today onwards learn it as an-10-inflammatory? Because that way you will always remember that anti-inflammatory is basically interleukin 10. So learn it as an-10-inflammatory. Easy? Okay. Let's go forward. Which is the interleukin that helps in T cell maturation? And the answer to that question is going to be interleukin 7, because the way you write 7, you can just make a T in front of it. Life will be easy. So repeating, the way you write 7, you can make a T in front of it. So interleukin 7 helps in T cell maturation. Which is the interleukin that helps in Eosinophil activation. Fifth alphabet, guys, interleukin, which helps in eosinophil, A, B, C, D, E. So the fifth alphabet is interleukin 5. Next, which is the one helping in neutrophil recruitment? This I just now taught you. I'll have to take you back to a table. Do you remember which was the interleukin which was helping in neutrophil activation? I can see for neutrophils I had an interleukin called interleukin 8. I just have to write that down again now. Interleukin 8 is going to help in neutrophil recruitment. So now let's read the story again. When I say fever, the main one for fever is going to be interleukin 1. That's the main hero that we have over here, interleukin 1. TNF alpha also causes fever, but N and A, no appetite, that is more famous. Which is the one that can act both ways, pro and anti? 4 and 6 can act pro and anti. Which is the one which is only anti? You will learn it as AN10. Very, very important. You will learn it as AN10. So interleukin 10 is the only one which is only anti-inflammatory. Which is the one that is going to help in T? So from 7 you are going to make a T. So interleukin 7 helps in it. Which is the one for eosinophils? Eosinophil 5. So interleukin 5. And what is for neutrophil? Interleukin 8. The story is obviously not over, because now I want you to focus on something, because what happens in the paper is you are in a hurry and you're sometimes very excited that your mnemonic has come. So you read eosinophil and then you forget to read what beyond eosinophil is written. Guys, if I would have asked you eosinophil recruitment, attracting an eosinophil, if I would have asked you attracting an eosinophil, how do you attract? Attract means you're going to get an eosinophil. Eosinophil, getting it in a taxi. That is eotaxin, okay? So eotaxin and interleukin 5 are totally different. Both are for eosinophils, but for different things. I'm telling you once again, eo means eosinophil, taxi means getting it in a taxi. So if I want to get an if I'm sitting here, I want to call an eosinophil, how will I call it? I will call it with eotaxin. But if I want to activate an eosinophil, if I want to switch it on, what will I do? Then I will use interleukin 5. Don't get confused. Calling it in a taxi, you use eotaxin. Activate it, switch it on, that is going to be interleukin 5. Interleukins are not over, because now I'm going to tell you which is the interleukin which regulates acute phase reactants. Firstly, learn it is interleukin 6, and I'm going to correlate it with the life changer for all of us, that is COVID also. But before that, you need to know what is APRs. APR stands for acute phase reactants. APR stands for acute phase reactants. And please know that I can see something over here, that some things are known as positive APRs and something are known as negative APRs. Means what? See, for example, today someone got fever. You must have seen in COVID, everyone used to get fever. And you remember in COVID, when we used to do someone's blood test, you must have seen in COVID patients, C-reactive proteins used to come very, very high. Ferritin levels used to come very, very high. Fibrinogen levels used to come very, very high. So basically, not just COVID, whenever someone has any kind of fever, there are some things which will increase in the blood. So remember, if they increase by more than 25% of the normal, that is known as a positive APR. And some things in the blood on having fever, they will go down. So if they decrease by 25% of the normal, they are going to be negative APRs. Repeating, if they increase by 25% from normal, this is the baseline. All the molecules are at baseline. The things which will increase by 25% in fever, I'll call them positive APRs. The things that will decrease by 25% of the baseline are negative APRs. Now, so many things are positive, I can't learn the list. It's very lengthy. C-reactive, everyone knows the full form of C-reactive protein, right? So CRP, C-reactive protein. Then we have ferritin, then we have hepcidin, we have haptoglobin, we have fibrinogen, we have procalcitonin. SAA, for those who don't know what is SAA, it stands for serum amyloid associated. You will be studying this under the amyloidosis chapter, okay? Serum amyloid associated. So basically all these things increase. I can't learn so much. Let me on the other hand learn what is going to decrease. So that makes my life a lot easier. So I have learned the mnemonic as TAN. That is the best mnemonic that I can think of. So I realize all these trans things. The three trans means transferrin levels decrease in the blood when we have fever. Transcortin levels decrease in the blood when we have fever. Transthyretin levels decrease in the blood when we have fever. So whenever we have fever, all the trans things are going to decrease. Okay. Other than that, A and A. So one A is albumin, and if you can learn the other A, that is antithrombin. That is antithrombin. No compulsion on learning this. If you can comfortably learn it, learn antithrombin. Otherwise, it's not important. So albumin and antithrombin levels decrease. And what is N? This is a mnemonic for which APRs? For the negative APR. So what is TAN? Trans. Transferrin, transcortin, transthyretin, A and A, albumin and antithrombin. And finally, these are negative. So I've learned the negatives means these levels are going to decrease in the body, in the blood, and all the others become positive. Their levels will increase. But who decides? Who decides that what will increase and what will decrease? It is interleukin 6 who is going to regulate this increase and decrease. That is where, if you remember in COVID, what was the most important interleukin which was creating that cytokine storm? It was interleukin 6. So interleukin 6 was going up, and that is why all these CRP, ferritin and everything was going up. Now that correlation is happening. Before we move forward, I want to ask you all a question. Do you guys know what is CRP? If I ask you what is the full form of CRP, all of you will very nicely just end up writing C-reactive protein, but that's still not the full form, right? What is the full form of CRP? Means what is the C in CRP going to stand for? C stands for the carbohydrate or the capsular polysaccharide of pneumococcus. So C alone has the biggest full form. It is the capsular polysaccharide of pneumococcus. That is what you need to know. So they will ask you this question, that what does the C in C-reactive protein stand for? So either you can call it the carbohydrate or you can call it the capsular polysaccharide of pneumococcus. So coming back, I hope everyone is clear with their interleukins, which was most important. So I have finished off number 1 with the interleukins. We have finished with the TNF alpha. Now we are left with the last variety, and that is interferon. So let's start discussing. For that, you will have to actually go ahead and tell me the types of interferons. And this interferons is equally important in when you go to virology, in microbiology. Over there also interferons are going to come. I'll tell you why are they important in virology. So remember, when we talk about interferon, interferons are of 3 types: interferon alpha, interferon beta and interferon gamma. So first thing is, you will have to know what is their source. Where are they actually going to come from? So interferon alpha, beta, gamma come from LFT, liver function test, LFT, but it's not liver function test here. L stands for leukocytes. L stands for leukocytes, particularly macrophages. F stands for fibroblast. F for fibroblast, and T for T helper 1 cell, which is a very famous previous year question. So repeating, LFT, leukocyte, fibroblast, T helper 1 cell. First have I learnt where are they coming from? What is the... Now I'll have to tell you what are they doing. So what is the function that we have? So please remember alpha, beta, gamma, and the function is also going to be alpha, beta and gamma. That's the best way of learning it. A means what? It is going to have antiviral properties. It is going to have antiviral properties. What is B? B is going to be used in the treatment of a B for brain disorder. Which brain disease? Multiple sclerosis. So in the treatment of multiple sclerosis, interferon beta is used. And what is G? So remember G for G. There are 2 things with G that I'm going to tell you. Number 1, please remember, interferon gamma helps in granuloma formation. Interferon gamma is going to help in granuloma formation. And next G is, it is used in the treatment of a disease which has granulomas. What is the disease? Chronic granulomatous disease. That is why I've learned that interferon gamma, G for G. Number 1, it is going to help in granuloma formation. Secondly, it is used in the treatment of a disease that has a granuloma. So let's do a very quick recap, because this is the best flashcard slide that you can have for interferons. Let's quickly revise. When I'm talking about interferons, they are alpha, beta, gamma. What is the mnemonic for the source? LFT. They come from leukocyte, fibroblast and T helper 1. What is the mnemonic for their functions? Same. Alpha, beta, gamma. Functions are A, B, G. A means it has antiviral properties. That is why I will teach you in virology. It is being used as a lot of antiviral topical creams if you want to fight against viruses. Interferon beta, treatment of a brain disease. Which one? Multiple sclerosis. And interferon gamma, G for G. It is going to help in granuloma formation, and it is going to be used in the treatment of chronic granulomatous disease. That is what you have for interferons. Having said that, we are done with all the cytokines. Interleukins, interferons and TNF alpha is done. Now we move on to the last variety that we have in newly synthesized mediators, and those are the arachidonic acid metabolites. So first and foremost, you need to tell what is AA. AA stands for arachidonic acid, and this is something that you will read in path, in micro, in biochem, in pharmacology and in medicine. This is a 5-in-1 kind of a topic. Everywhere you're going to read the arachidonic acid metabolites, and I'm going to teach you from all points of view in one go only, so that once you read it and you don't have to read it over and over again in different subjects. So what is arachidonic acid? Firstly, remember arachidonic acid has 20 carbon atoms. It is a 20 carbon compound. The first question which will come to everyone's mind is, where is arachidonic acid coming from? What is going to result in the formation of arachidonic acid? So please remember, it comes from phospholipids, and it comes from very essential phospholipids, essential fats like linoleic acid. It comes from phospholipids. So imagine you have to break phospholipids. What enzyme are you guys going to use if you want to break phospholipids? Either I will use phospholipase A2, or I am going to use phospholipase C. And that's a famous question in a lot of your Q banks and a lot of your PYQs and GTs. Which are the two phospholipases that break the phospholipids? Phospholipase A2 and phospholipase C. Arachidonic acid is formed. How many carbon atoms does arachidonic acid have? 20. After this, what is going to happen? I am not interested in arachidonic acid. I am interested in the arachidonic acid metabolites. What is going to form out of it is going to cause inflammation. So remember, this arachidonic acid can then enter into 4 pathways. I know all of you have read 2 main pathways always, that is COX and LOX, but I'm going to make your life a little more complicated by telling you 4 pathways. But yeah, all your concern has to be on COX and LOX. 4 pathways will be, the other 2 will be just quickly going through one line of it. So I'll quickly get you familiar with it. There is a cytochrome P450 epoxygenase pathway. Cytochrome P450 epoxygenase pathway, and there is something known as isoicosanoid pathway. Don't get too worried about it as to why am I using such tough names. Everything's gonna be fine. You just need to know one line. Your main focus will always be on the COX pathway and the LOX pathway, and that is what I'm starting with. I'm first starting with the COX pathway. What is COX pathway? COX stands for cyclooxygenase. Cyclooxygenase. So please remember, for example, here you have the arachidonic acid, and on arachidonic acid you have acted 2 cyclooxygenases. There is something called cyclooxygenase 1 and there is something called cyclooxygenase 2. I'll talk about their different types, but you know what is formed because of the cyclooxygenase pathway? We have the formation of PGs. What is PGs? So coming back, because of cyclooxygenase, we will have the formation of PGs. PGs means prostaglandins. That is the first thing that they are going to ask you in the paper. Which is the pathway responsible for prostaglandin formation? And that is PG COX pathway. So the first prostaglandin to form, remember, is G. The first one to come in the story is PGG2. After G, guys, what is the next alphabet? The next alphabet is H. So the next one to form in the story is going to be PGH2. And after H, I realize H for whole spectrum. H means whole. Everything is going to come. The alphabets before that, the alphabets after that. What were the alphabets before that? So I missed out on, I started with G, right? So G and H. What about the D, E, F? So this means this is going to give rise to PGD2. It is going to give rise to PGE2. It is going to give rise to PGF2 alpha. So D2, E2, F2 alpha. The story is not over. It also gives rise to the future alphabets. What are the future alphabets? After H comes what? After H comes I. So it is also going to give rise to PGI2, and please remember PGI2 is also known as prostacyclin. PGI2 also known as prostacyclin. Other than that, it gives rise to TXA2, which is also known as thromboxane A2. It is also known as thromboxane A2. Let's do a recap. It's very, very easy to learn. Which is the first prostaglandin formed? The first one is PGG2. After G comes the next alphabet H, and H for whole. Everything is going to form after it. All the previous ones also, D, E, F. All the future ones also, that is I. I is known as what? Prostacyclin. And then there is a weird sounding one called thromboxane A2. Should you go ahead and do their functions? Yes. Now note, PGD2, D for dilation. What does PGD2 cause? It causes vasodilation. D for D, vasodilation. What does PGE2 cause? E2 causes pain. How do I learn it? If I pinch you, if someone pinches you, or whenever you experience some kind of a pain, what are you going to scream out? E is the sound that you take out. So E means, oh, I'm feeling a lot of pain. So E is going to be for pain. And what is F2 alpha? I think you all know there is a treatment drug in gynecology by the famous name of carboprost. You all know that carboprost that helps in uterine contractions and cervical ripening. All of that is nothing but PGF2 alpha. So that is the use of F2 alpha. So D for dilation, E for E sound, that is pain, and F2 alpha is a drug that you use in gynecology. So it's carboprost. Now coming to the others. Prostacyclin and thromboxane A2. Let me tell you, learn one of them. The other one is going to be the opposite of it. So one of them you have to learn, that is your choice. So please remember, let's try and focus on thromboxane A2. Okay, let's try and focus on thromboxane A2. So I realized thromboxane A2 has A in it. So something with A is going to happen. And prostacyclin has PGI2. So something with I is going to happen. So if they ask you, which of these is going to aggregate the platelets? Which of them is going to aggregate the platelets? Thromboxane A2 will aggregate the platelets. On the other hand, if they ask you who will inhibit platelet aggregation, I will inhibit platelet aggregation. So see, they're opposites. Now, thromboxane A2 aggregates the platelets, brings them together, aggregates. PGI2, I for inhibition, it does not aggregate the platelets. Now, when I said thromboxane A2 aggregates, means it brings everything closer. What is it going to do to the blood vessel also? To the blood vessel, it causes vasoconstriction. This means prostacyclin will cause vasodilation. So simple, thromboxane A2 is all about bringing things closer. It's all about aggregating. Platelets are also aggregating. The blood vessel is also constricting. PGI2 is inhibition of all of this. PGI2 is going to open up the platelets, not let them aggregate, open up the blood vessel, vasodilation. That is how you learn this. And I think this entire story has been sorted. Okay, now the story is almost sorted if you finish the pharmacology aspect also along with it. So everyone, please listen to me. Whatever pharmacology you have read up till now, you can read up till now, let me finish it off. Did I tell you that the first enzyme which started off this entire story was phospholipase A2 and C? And if you stop this pathway at that level, nothing is going to move forward. So either you can stop this pathway at the phospholipase level, or you can stop the pathway at the COX level. So let me talk about both of them. So like I said, that there was a phospholipid, and phospholipid was acted upon by phospholipase A2 and phospholipase C to get arachidonic acid. Arachidonic acid was then acted upon by COX-1 and COX-2 to get prostaglandins. That's the story that we've done up till now. Okay. If I want to stop it at this level, I want to stop phospholipase A2 and C, and I don't want this pathway only to start, then you give the wonder drug, which is steroids. What is the function or the mechanism of action of steroids? It inhibits phospholipase A2 and C. So the process will never start because of steroids. Versus how do I inhibit COX? I think everyone is aware of the fact. First, let me tell you that COX-1 and COX-2 may say one of the COX is for this inflammation, but one of the COX, remember COX-1 is a good person in your body. COX-1 is a gastroprotective enzyme that God has given you, something that is constantly in your stomach and protecting your stomach, that is COX-1. Now, you know the drug that can inhibit both of them? The drug that can inhibit both of them is going to be NSAIDs, for example, aspirin. I think everyone is aware that aspirin is going to shut off both COX-1 and COX-2. Both are going to be shut off. But in that process, have you done a mistake? If you have shut off both, means you have shut off COX-1 also, means you have shut off your protector. You've gotten rid of your protector. What is the stomach going to suffer with? The protector is gone. The stomach is going to have ulcers. Don't you study? One of the side effects of aspirin is going to be peptic ulcer disease. Why? Because you have gotten rid of the protector over here. Is that a good idea? Not a good idea. Let the protector be. Don't touch or play with nature. Don't touch what God has given you. So let the protector be. You just shut off the other one. That will be a wiser decision. So now they are saying that what we prefer is only shutting off COX-2. Selective COX-2 inhibitors is what my preference will be. And I think everyone knows all the COXIBs. For example, we have celecoxib, and all the COXIBs are going to come under only the COX-2 getting inhibited. Right? So let COX-1 function. Don't play with it. Let that protect your stomach. You focus only on COX-2. That is the entire pharmacology about it. Let's move on further, because up till now I have only spoken about one pathway, and that is the COX pathway. Now I'm going to go ahead and talk about the LOX pathway. And you know what is going to be formed in LOX pathway? LT-LX. What is LT-LX? Let's talk about it. Look at this, guys. I have the same arachidonic acid, but now it will be acted upon by LOX. You need to tell me what is LOX. LOX is the full form or is the short form of lipooxygenase. Now I'm using lipooxygenase, and please note that I can use something known as two mechanisms or two pathways. There is something known as 5-LOX, 5-lipooxygenase, and there is something known as 12-LOX, 12-lipooxygenase. What's the difference? Remember, the 5-lipooxygenase is going to form LTs, that is leukotrienes. Leukotrienes, okay? And now you will ask me which, remember we did prostaglandins. Which was the first prostaglandin to form? PGG2, GHI. That is how it was going. Here the story is very simple. We start with A. The first alphabet is what we are going to actually start with. So remember, leukotriene A4 is what I am starting with. From A, what will form? From A, the entire world will form. From leukotriene A4, I will have leukotriene B4. And from leukotriene A4 only, I will have leukotriene C4, D4, E4. Simple here. The story is so simple, na? From A only everything is formed. From A you have B, from A only you have C, D, E. Have I taught you leukotriene B4 somewhere very shortly, very short time ago when we were doing acute inflammation events? Did we study leukotriene B4? Remember LIC mnemonic, chemotactic agent, leukotriene B4L, leukotriene B4 tha. So that is the chemotaxis. It is going to help in chemotaxis. Okay, what about C4, D4, E4? This is something you guys will be reading in your medicine and pharmacology. They are also known as SRSA. What is SRSA? It is slow reacting substances. They are slow reacting substances. They act very, very slowly, but what can they cause? A for anaphylaxis. They are slow reacting substances of anaphylaxis. See, SRS for slow reacting substances, A for anaphylaxis. Anaphylaxis means something allergic, something deadly, and this means that they have a possibility of going on and causing asthma. Did you all realize that when you were studying the pathophysiology of asthma in medicine, C4, D4, E4, you must have studied that they are the ones which are going to cause bronchoconstriction. Bronchoconstriction basically means that they are going to result in the formation or the development of asthma. Yes. So did I tell you some hero which was also causing asthma? Histamine, serotonin, the hero obviously has to cause, that was also causing asthma. So if I ever get a question, and this you will find as a question, if I ever get a question that is histamine the main person who's causing asthma, or are leukotrienes more potent, be it very, very clear over here. I want to make it very, very clear. Leukotrienes are more effective in bronchoconstriction than histamine. They are more effective in causing bronchoconstriction. So that is another thing that I kept telling you. If you compare it with histamine, histamine is just like that blockbuster hero. Hero means he is the one who is showing off everything, but who is doing all the work behind the scenes? other people who are working and going back home, not showing off, not taking the limelight. So although histamine is most spoken about, we are coming to a conclusion gradually that other people are more potent. So who causes more bronchoconstriction than histamine is actually the leukotrienes. Leukotrienes C4, D4, E4. Coming back, all of this was happening because of 5 LOX. Now my story is what happens because of 12 LOX. Because of 12 LOX, we have the formation of LX. LX means lipoxins. Lipoxin A4 and lipoxin B4. And you know what are they? They are anti-inflammatory. They are the ones which are not going to result in inflammation, which are going to stop the inflammation. Okay, so remember, that is anti-inflammatory. Well, having said that, we have finished the LOX pathway also, so it's a must that why don't we go on and do the enzymes also? Why don't, or you know, when we do this, why don't we do the pharmacology inhibiting these enzymes? So yes, let's quickly study that. Everyone note, I'll take you back to the pharmacology slide that we had. Now, we've done steroids, we've done the COX inhibitors. Now I know that the same arachidonic acid could have taken the LOX pathway and resulted in what all? So, resulted in the formation of leukotrienes, and I'm particularly worried about those asthma leukotrienes. That is leukotrienes C4, D4 and E4, the asthma causing one. So, what drugs can I use? Number one drug, so if I take you back to the pathway, number one drug that I can use will inhibit this. The first enzyme is going to chuck off, and that is going to be xylutin. Please remember, it is going to be xylutin. Xylutin is going to cut off the first step and the story ends. Before it starts, the story ends. But you tell me, if I cut it off at the first step, who was causing asthma? Asthma was caused by C4, D4, E4. Target them now. By cutting off this, you are even cutting off leukotriene B4. Leukotriene B4 has no role in asthma. Why are you cutting him off unnecessarily? Not a good thing to do. So xylutin is just stopping everything which is not required, right? So then they realized that better than xylutin, pick up something which is only going to stop the C, D and E. And those are leukotriene receptor antagonists. Everyone, please remember this, that we have a set of drugs called, which most of us have consumed, have prescribed, leukotriene receptor antagonists, because they are going to be against the receptor of which leukotrienes? The main culprits, C4, D4 and E4. And what are the names of these drugs? They are going to be montelukast and zafirlukast, and many more of this family, of course. So montelukast and zafirlukast, all these leucast are the leukotriene receptor antagonists. Montelukast and zafirlukast, they are going to go and inhibit only these set of molecules. So remember, again you can write over here, leukotriene receptor antagonists are going to be given. I know it's become messy slide, but I hope this will help you figure out everything. Let's do a recap. What is the drug that inhibits the full enzyme only? The drug that is going to inhibit the enzyme is going to be xylutin. But which is the drug which is going to inhibit only these, which are the leukotriene receptor antagonists? They are going to be the montelukast and the zafirlukast. So this is your full pharmacology correlation. A quick recap. Please remember, if you want to stop everything on the first step, steroids will stop phospholipase A2 and C. If you want to stop all the COX, aspirin is going to stop everything, but is deadly or is very dangerous because it can cause peptic ulcer. So let COX-1 be as it is, let it function, just focus on COX-2, just break down COX-2, that is by the COX-IBs. When you come to the LOX pathway, either you can stop it at the first go, that is xylutin, or you will stop it at the leukotriene receptor antagonist, that is the main asthma causing one, C4, D4, E4, that is by montelukast. That finishes off all of your arachidonic acid metabolites also. But if you all remember, going all the way back, I had told you the main people to focus on are COX and LOX, but you are also going to have some minor things like cytochrome P450 epoxygenase pathway and isoicosanoid pathway. So very quickly I'm going to wrap up on these two things. So listen to me very carefully. I have the same arachidonic acid, but the name of the pathway is clear now. What is the name of the third pathway that we have? Cytochrome P450. Cytochrome P450 epoxygenase pathway. So which enzyme is going to act? Cytochrome P450 epoxygenase will act. This will result in the formation of two molecules. There is something called EET that is formed, and there is something called HETE that is formed. Guys, please don't try to go into the full forms because they are really complicated. It is something called epoxy icosa tetraenoic acid. If you want to go into it, I don't think you want to, right? So there's something called EET and there's something called HETE. Just remember, one of them causes hypertension, one of them does not cause hypertension. So as simple as that, when you're talking about H, this is going to cause renal vasoconstriction. This causes renal vasoconstriction. Anything that will cause vasoconstriction means it is going to cause renal hypertension. So simple, H for H. H for H. HETE causes hypertension and EET is going to be antihypertensive. Antihypertensive. So you know the only purpose of me telling you this pathway was that EET, because it is known to decrease the patient's blood pressure, they are now trying that they should make a drug out of it because it will help in the treatment of hypertension. That's the only purpose of me telling you. So two molecules. The name of the pathway, cytochrome P450 epoxygenase pathway. So cytochrome P450 obviously becomes the enzyme. Two molecules form EET, HETE. Which causes hypertension? H for H. HETE causes hypertension. EET does not cause hypertension. Simple. The last pathway that we have is the easiest. This time, the arachidonic acid is going to be acted upon by which enzyme? So see, in the first pathway, it was COX enzyme. In the second pathway, it was LOX enzyme. In the third pathway, it was some weird cytochrome P450 epoxygenase. The fourth pathway is finally nothing to do with enzymes. Non-enzymatic pathway, and this results in the formation of isoprostanes. That is why what was the name of this pathway? Isoicosanoid pathway. Even if you want to learn it as the isoprostane pathway, good to go. And why am I reading about this in inflammation? This causes vasoconstriction. This is causing vasoconstriction. So simple. Now have we studied all the four pathways and the entire story is done? COX, LOX, cytochrome P450 epoxygenase. So three enzyme related pathways. And this isoprostane one is nothing to do with enzymes. It's a non-enzymatic pathway. With which, guys, you realize that your newly synthesized mediators finally come to an end. Nitric oxide, platelet activating factors, chemokines, cytokines, arachidonic acid metabolites. Obviously, all of them come to an end, and that means you are ready for which family? Your cell-derived family is done. Move away from the cells. Now you're going to go into the plasma-derived. And do you remember what all was there in plasma-derived? The K sounding ones. The plasma-derived had K sounding ones, the complement system, the clotting system, and the kinin system. And that is exactly what we are going to start with. We are going to start our journey with number one, the complement system, the first K. Now, I could have called it complement pathway also, but it's not one pathway, it's a system. It's a system of three pathways. So please remember, there is something known as the very, very famous classical pathway. There is something called classical pathway. Then there's an alternative route. So there is something called alternate pathway, right? There's some alternate route of starting it, alternate pathway. And then there is something called MBL pathway or lectin, or in other words, if you want the full form, mannose binding lectin pathway. Mannose binding lectin pathway. So simple, classical pathway, alternate pathway, and MBL pathway. Let me tell you one thing, no matter whichever pathway you start, your end result is going to be a molecule called MAC. I'll tell you what is MAC. So first you'll say that, okay, if everything is the same in between them, then why are there three pathways? Because their starting point, what are they activated by, that is going to make a difference. What are they activated by? So number one, the classical pathway is activated by immune complexes. What do you understand by immune complexes? Immune complex means antigen-antibody reaction. And which antibodies are going to come over here? IgM and IgG. Any immune complex that contains IgM and IgG will go and activate which pathway? The classical pathway. Talking about alternate, A for A, any immune complex containing IgA is going to go and activate the alternate pathway, A for A, simple. And when I talk about mannose, mannose is something in the bacteria. So any pathway which is directly activated by the microbial organisms Any pathway directly activated by the microbial organisms, that is going to be the mannose pathway. So simple, end result of everything is going to be the same, the starting is different. So classical by classical complexes, M and G complexes, alternate by A, IgA antibody complexes, and mannose by the bacteria or the microbial. Now let me show you what is the tough part going to be over here. So first and foremost, these are the three pathways which I've tried to put up over here. I'm going to tell you a shorter version of it, and then obviously I'll be telling you a detailed version of it. So let's start with the shorter version first. There is something known as the classical pathway. How do you activate the classical pathway? By immune complexes, by antigen antibody complexes. And can you see which are the set of molecules that you have activated first? The first molecule that I activated was C1. After that, I have a rule. First go for even numbers, then go for odd numbers. C1 will then activate all the even numbers, that is C4 and C2. They will get activated. After you have activated C4 and C2, what is the next number that you are going to activate? The odd numbers. So then you go for 3. After that, you go for activation of 5. That's a shorter way of learning it. So you start from C1, then you activate the even numbers, that is 2 and 4, and then you go into the odd numbers, that is 3 and 5, and half of your pathway is over. I want to give you obviously a complicated version of it. So let's start. When we start with the classical pathway, guys, obviously there was some antigen antibody complex that started it, and the first thing to get activated was C1, which formed C1q. Okay, QRS, many times it formed. I am interested in C1q. Okay, this C1 then went and activated C4 and C2. Now I just want to tell you one offhand thing. Whenever I'm saying something is getting activated, means it is forming an A version and a B version. Some of the things, some of the A's and B's I'll keep, some of the A's and B's I'll discard. Okay, but when I say something is getting activated, means it's being broken into A and B. So when I'm saying C4 is getting activated, means it's getting activated into A and B. Similarly, C2 is getting activated, it is getting activated into A and B. That's your basic concept. Okay, so remember, C4, I want to keep the 4B 2A. So how are they activated? They are activated into C4B2A. So now 4 activated, 2 activated, even numbers done. Move on to the odd numbers. Which is the one that you will activate? You will want to activate C3. So can I say who is activating C3? 4B and 2A is activating C3. Can I call it the C3 convertase? Can I call it C3 convertase? So C4, that is what was there in the previous one also, if you realized. They were together known as the C3 convertase because they were converting C3 into what? They were converting, again, when I say someone is getting activated, means there's going to be an A and there's going to be a B. This means C3 is now going to combine to this molecule. So what do you have? You have 4B, 2A, 3B. So BAB, alternate chalega. 4B, 2A, 3B is formed. 3 has been converted. 3 is a part of this complex now. After 3, what's the next odd number? The next odd number is C5. So can I say these 4, 2 and 3 that are converting C5 are now known as C5 convertase. And when I say it's getting activated, means C5A and C5B. I am now going to leave all the past 4B3, everything I'm going to leave. Now I'm only going to pick up the C5B. And you know what am I going to combine it with? I'm going to bring all its friends together. 6, 7, 8, 9. I'm going to pick up the C5B, combine it with the next 6, 7, 8, 9, and together they are referred to as the membrane attack complex. What is membrane attack complex? Membrane attack complex, whose membrane are you wanting to attack? The bacteria. I want to kill the bacteria. So is this going to go, and for example, if this is a dirty dark bacteria over here that I have made, is this going to go and break the bacteria? Yes, it is going to cause bacterial lysis. Whose membrane is it attacking? The bacteria's membrane it is going to go and attack. It's not a very easy complement system. You read it in weather path or micro, it's tough, but we'll try to simplify it. Let's repeat. The story starts off with C1. It has to start with number 1. First, I'm going to target all the even numbers. Then I'm gradually going to go and target all the odd numbers. So first I activate 4 and 2. So C4B2A is formed. That is now going to activate 3. So then what we have is 4B, 2A, 3B. That is now going to activate 5. And as soon as 5 is activated, 5B, 6, 7, 8, 9 are going to form the membrane attack complex and kill off the bacteria. Okay, having said this, coming back, you realize that your classical pathway is finally a tick mark. Let's talk about the alternate pathway. Alternate pathway is not going to start from that one and all those usual. Alternate means it's going to take some alternate route. It is going to start from C3. That's why it's alternate. It's taken some alternate route. So C3 along with a lot of factors, factor B, factor D, properdin, it is going to break. So please remember, I'll try to fit in the alternate pathway over here only. When I talk about the alternate pathway, the story starts with which antibody? With IgA, and it is going to start with C3. So please remember, C3 is going to be broken into 3A and 3B as always. Every molecule is A and B. So this 3B is then going to combine. This 3B is then going to combine with something known as factor B. Please listen to this very carefully. Factor B, factor D, and properdin. There is involvement of factor B, factor D and properdin. And you know what happens? Especially factor B. Factor B also starts breaking like these numbers into BA and BB. So you know all the B's come together. I have something known as 3BBB. The 3B from here and BB from here, they come together and they make a complex called 3BBB. And you know whom are they going to go and break? Again the C3. They basically want to come and merge with this pathway at the C3 level. They again go and merge at the level of C3. So first C3 broke. Let's do a recap. First C3 broke into C3B. It took care of all the B's together. It's taken all the B's of its family 3BBB and has gone and attacked another C3. So now ultimately what is going to happen? If it attacks the C3 once again, is it going to result in the formation of 3BBB, 3B once again? If it's again going and attacking the C3, means it is once again going to form C3B, BB, 3B, and finally after that, that will go and attack your C5. And once C5 is attacked, it's going to be the formation of the membrane attack complex. You can have a look at it once again. Let's have a look at it again, guys. We started off our story with what? We started off our story with 3B. 3B acted upon with factor B, factor D, properdin, 3B, BB bana. That has gone and again acted upon on C3. So means C3B, BB, 3B. And once that complex is ready, that is going to go and act on C5. Once C5 is done, I have the membrane attack complex ready. That was the alternate pathway. Now I'm only left with the mannose binding lectin pathway, and that is neither going to start with C1 nor is going to start with C3. If I see, this is starting with the middle molecules and directly going at C3 convertase, means it's starting off with molecules called C2 and C4 and is directly going and targeting where? C3 convertase. So if I realize now which is the meeting point of all these pathways, be it classical or alternate or MBL, they are coming and meeting at the level of this C3 convertase, means they are coming and meeting at the level of something where I want to break C3. And that means what is the most critical step that I can talk about in the complement system? Please remember, the most critical step is a very favorite question, and that is the conversion of C3. The C3 conversion is the most critical step because all of them are going to come and sit down over there. Okay, having said that, I have a couple of questions for all of you, which is based on whatever we have studied up till now. Nothing new, but whatever we have studied up till now, let's see if you are able to answer it. Can you remind me, out of all of these, if I ask you, what is the composition of membrane attack complex? That is what they will be asking you. What is the composition of membrane attack complex? It is C5B to C9. So 5B, 6, 7, 8, 9. Question number 1, sorted. What is the composition? Or what is the next question? Which are all of these which are chemoattractants? If you remember, we had one question on chemoattractants which are going to cause chemoattraction, and one question was there on opsonins, right? So now we need to know those. Please remember, for chemoattraction, you had the A molecules, particularly C5A, which is a very famous question. Then What else can the A molecules do? Any idea? What else can C5A, C3A, all these AA molecules, they are also going to be anaphylotoxins. They are also responsible for anaphylaxis. So remember, all the A molecules are anaphylaxis. All the A molecules are attractants, but C5A is the most potent attractant. And all the B molecules, C3B, 4B, 5B, all the B molecules were opsonins, right? Are we clear with this basic? Nothing new that I taught you, everything from what we studied. Now I'm going to teach you something new, because with this complement system, you also need to know all the diseases. So tell me, which is the most common complement deficiency known in humans, and that is C2. May or may not come across as a disease, but at least C2 is the most common complement deficiency. Okay, in someone, if the early complement proteins end up undergoing a deficiency, C1, C2, C4, that has been associated with an autoimmune disorder, that is SLE. That is associated with SLE. So early ones, SLE. If the late ones undergo a defect, that is C5b to C9, C5b to C9 was membrane attack complex, there is infection with many capsulated organisms, and one very famous infection is going to be Neisseria. Let me focus on this, very, very important, very famous infection that you need to know is Neisseria infection if there is MAC deficiency. Now comes the question that has come so many times in NEET, in FMG, in INI-CET. What is C1 inhibitor? So now, all said and done, everything was going, this pathway was going on and on and on, and in the end, membrane attack complex was forming everywhere, bacteria was being killed. Do you need something that should stop this pathway? Because if this keeps going on in my body 24/7, it will end up killing me only. It will end up killing my own cells. So when required, it should work, and when not required, it should not work. So they realized that you need some people to control this pathway. And that is when they came up with that if you do not let the first step occur, C1 is inhibited, then the entire pathway will not occur. So simple. If you want to control this pathway, do not let the first step occur. So for that, God has given us something called C1 inhibitor. C1 inhibitor controls the activation of C1. It controls the activation of C1. Basically, it controls the complement system. But now I'm talking about a disease when this controller is lost. If the controller is lost, then, like for example, if you are trying to get distracted, I am the one who's going to control you. But if I only go away, or if you switch off this app right now and send me off, then there's no control. Then you will be doing whatever you want. So if the controller is lost, big time problem. And that is going to be hereditary angioedema. That's the name of the disease that you all have to learn. It results in hereditary angioedema. And please guys, the way I've said edema, first thing you need to know, where is the edema going to present? So the edema is most commonly present around the eyes and the lips. So let's draw a funny looking diagram. So if this is a person, for example, and if they ask you where is the edema going to be, so the edema is going to be a weird swollen kind of an eye. Number one, the edema is periorbital, and second, the edema is going to be around the lips. So a very weird looking man that we had drawn, right? So remember, there is a periorbital and a mouth or around the lips. That is where the edema is there. All that is okay. When can this come as a medical emergency? Because that is a question that comes. What if the person ends up having in the neck, means larynx? If someone ends up having laryngeal edema, then this means he will not be able to breathe. If there is laryngeal edema, this means the patient has entered into respiratory distress, and that makes it a medical emergency. Run to the hospital. So remember, if the edema is around the eyes and lips is still understandable, but if it occurs in the larynx or the respiratory tract, big time medical emergency. That is what has come in the question a lot of times. So remember, hereditary angioedema is because when the controller is lost, C1 inhibitor is deficient. When you're talking about this, why don't you talk about the others also? Let's talk about all of these. And you know what are all of these? All of these are controllers. That is what I want to tell you. All these things that are mentioned over here, CD55, CD59, factor H, factor I, CD46, all of these are controllers of the complement system. So CD55 and CD59, what do they break? How do they control? They break the two most important odd number converters, C3 converters and C5 converters. So if these three are broken or these two are broken, complement system will not work. So they're good for us. CD55 and CD59 control your system. But just imagine if CD55 and CD59 go missing, the controllers are gone. Does it remind you of any disease in the blood? There is a disease called PNH, which means there is paroxysmal nocturnal hemoglobinuria. There is paroxysmal nocturnal hemoglobinuria that you have to know. Okay, next let's look at factor H, factor I and CD46. They are also controllers, but when factor H, factor I and CD46 go missing, it's a very important disease in medicine and pediatrics. That is going to result in atypical hemolytic uremic syndrome. Atypical hemolytic uremic syndrome. Finally, I want to correlate this with ophthal also. Have you guys studied that factor H is associated with something? Factor H defect in ophthal, what can you think of? Have you heard of ARMD, that is age-related macular degeneration? Age-related macular degeneration, that is because of factor H defect. So this is basically trying to tell you all the relations of complement systems that are going to come in the future in your life, be it in path or be it in other subjects. So remember, CD55 and 59 are PNH, factor H, factor I and CD46 are atypical HUS, factor H in ophthal is age-related macular degeneration. And for the time being, you just need to know that the same disease I will be telling you under, or the same set of things I will be telling you under kidney. Because now there is a disease called MPGN, which you have to know, which is very, very important. So please remember, for the time being, I can only tell you a little bit about it. MPGN type 2 is now said to be a complementopathy. It is said to be a complementopathy, means there is a problem in the complement system. What is MPGN? It's a disease in the kidney, membranoproliferative glomerulonephropathy. Membranoproliferative glomerulonephropathy is a defect in the kidney. So right now just know it's something to do with complement system, okay? Having said that, the complement system is done. What is the next pathway that you should be knowing? And that is going to be the next K, that is the clotting system. Right now I'm giving you a very brief overview of the clotting system, because this is a very, very detailed topic that you are going to study when you go to platelet chapter. It's like a lot of detail. Right now I'm giving you the best possible summary over here, okay? So when we talk about clotting system, it is basically to do with blood clots, right? Basically to do with blood clots. Now you'll suddenly feel that I was in the chapter of inflammation. Why has ma'am deviated the entire flow and taken me towards blood clot all of a sudden? I'll link that also. So talking about clotting system, how is blood clot formed in the body are two pathways. One is going to be the intrinsic pathway and one is going to be the extrinsic pathway. And I know all of you have been struggling with learning these pathways since a very long time. So we are going to try and simplify it. So when I'm talking about intrinsic pathway, and I'll tell you, no matter how you'll start this pathway, you will come down to getting it common at factor 10. Because whatever will happen after factor 10 will then become the common pathway. Both of them are going to merge at factor 10 and that will become the common pathway. So let me tell you, the mnemonic for this table is 10 for 10. 10, 10, 10. Everywhere I'm going to talk about 10, means intrinsic pathway is going to be 10 for 10. What does that mean? Which is the first factor to get activated? 12. There is activation of 12. First is 12 gets activated. After 12, what gets activated? Factor 11 is activated. After factor 11, what is activated? 9 is going to be activated, and 9 has a cousin brother next to it, which is factor 8, and they are going to activate together 10. So isn't it 10 for 10? Let's repeat. 12, 11, so 10 you can write as 10E10. Because you're talking about so many 10s, I've written it as 10E10. 10E10 means 12, 11, 9, 8 and they activate factor 10. Here factor 10 is activated. Simple. So easy to learn, right? 12, 11, that's the order. 12, 11, 9, 8, 10. 10E10. What about extrinsic pathway? In extrinsic pathway, there are two factors, factor 7 and factor 3. Guys, what is 7 plus 3? 7 plus 3 is also 10. So in the extrinsic pathway, when these two factors get activated, I hope all of you will know that they are going to go and activate factor 10. So all in all, from both places, I have activated factor 10 using 10E10 for 10 and 7 and 3 for 10. Ho gaya na? Easy. 10E10 for 10 and 7 and 3 for 10. You've reached a common point. After this, just keep doing half, half, half. So first tell me your mathematics. Let me test. What is the half of 10? The half of 10 is 5. What is the half of 5? 2.5. But 2.5 can't exist. So I'll have to take factor 2. And what is the half of 2? 1. So now I have to do 10, 5, 2, 1, which means factor 10 in the presence of factor 5 is going to activate factor 2. Factor 2 getting activated. Do you guys know what is factor 2? So 10 was there, 10 in the presence of factor 5, that is going to be prothrombin. Prothrombin is going to get activated into thrombin, and please remember that after 2 is going to be 1, so this is then going to activate 1A into 1, and that is what is your blood clot. What is your blood clot? What is factor 1? Factor 1 is fibrinogen. Basically fibrin banega. From fibrinogen there is formation of fibrin that is going to occur, and fibrin is the blood clot that we are talking about. Fibrin is the entire blood clot that we are talking about. Let's do a quick recap over here, guys. We have something known as the intrinsic pathway and the extrinsic pathway. In the intrinsic pathway, we have 10E10. What is 10E10? 12, 11, 9 and 8 for 10. 10E10. What is extrinsic pathway? 7 plus 3 is going to activate 10. After 10, half. 10, 5, 2, 1. So 10 in the presence of 5 activates factor 2, activates factor 1. Your blood clot is formed. So if I asked you in a very simple method, what is the definition of a blood clot? You would have said blood clot is the last product formed, that is factor 1, that is fibrin. Now I want to ask you something more, and that is, all the way going back, is there any factor that I have not spoken about? So how many factors are there? There is 1 to 13 factors which we have. What all do you see missing over here? I don't see factor 13. So 13 I have to talk about. I don't see factor 4. I don't even see factor 6. So first, please remember, there is nothing known as factor 6 because there is no factor 6 existing. Factor 6 does not exist. So I cannot use it. Why didn't I use factor 4? Now I'm going to use. See, when you look at this junction point, the point of meeting, intrinsic is coming, extrinsic is coming, common is going, factor 4 is calcium. Let me tell you, factor 4 is calcium. So whenever you want to have something to do with junction, anywhere where the meeting and the exit points are there, calcium is going to be used. So intrinsic coming, extrinsic coming, going forward, that junction point, that Y, I'm going to use calcium over there. Calcium bhi use ho gaya, factor 4 also used. What am I left with? I'm left with factor 13. Where should I use factor 13? Because now, once I have the blood clot, you get something in life, then you get a little more greedy. You want something more. Now I've got the blood clot, but now we are becoming a little greedy. And I say, I want this blood clot to be very, very strong. I want a very strong and a stable blood clot. What should I do? I'll say, never mind, use factor 13. Use the last factor in the last step. You know what is the other name for factor 13? Factor 13 is known as fibrin stabilizing factor. Factor 13 is known as fibrin stabilizing factor and also given a very fancy name known as Lackey-Laurent factor. It is also referred to as Lackey-Laurent factor, and please remember this factor 13, the name only tells you fibrin stabilizing. It's going to look at the name, it is fibrin stabilizing. It's going to make your blood clot very, very strong. So please remember, finally, with the help of factor 13, my blood clot becomes really, really strong. Now coming back, have you used everything? So it's a very simple 10A10 for 10, 7 and 3 for 10, then halves 10, 5, 2, 1. And what are you left with? You're left with 13. So right in the end you use 13. That is how you learn it. Now, since I have taught you almost everything about it, let me also teach you what are the tests for the clotting system. So that now when we go to platelet chapter, I have pretty much nothing left for you to study more. What are the tests for the clotting factors? So you will say there are two pathways. One is the intrinsic pathway and the other is going to be the extrinsic pathway. So there are two tests that we have, and one is known as PT and one is known as APTT. So remember, for intrinsic pathway we use the APTT test, and the extrinsic pathway we use the PT test. I'm not telling you their normal values and details, that is for platelet chapter, but we always get confused which is PT, which is APTT. So remember, we are going to use the PET mnemonic. We are going to use the PET mnemonic. That is going to tell you that we have PT, prothrombin time. PT is for which pathway? PT is for the extrinsic pathway, and then APTT is for the intrinsic pathway. All said and done, your blood clot is formed. Why in inflammation am I studying this? Coming back to the main topic, why am I studying all of this in inflammation? Because the connecting link is here. Did you notice this thrombin? This thrombin is the one which can start inflammation. So just because of this one thrombin, I had to make you study so many things. But yeah, it's not a waste, because you know how tough this topic is and you know how 10/10 has made it simple, right? So remember, this is the one that is going to start inflammatory processes in the body. That is why we study it. And what were we doing? We are doing the plasma mediators. In plasma mediators, we had 3 Ks. Complement system finished, clotting system finished. What is the last system which is going to finish off everything for me over here, thankfully, is the kinin system. So you know the kinin system also you will understand if you have just understood the clotting system. Because do you know how did the entire story start? It started from factor 12. Remember 10E10, 12? The story started from 12. That same, I'm going to put a star mark over here so that when you revise in your notes, you should remember. This 12 is just not carrying out the blood clot formation. This 12 is going to go further and also talk about the kinin system. So you know when 12 is activated, what happens? When 12 is activated, please remember there is a molecule called prekallikrein. There is a molecule called prekallikrein which is converted into kallikrein, and after that prekallikrein is converted into kallikrein, this kallikrein converts kininogen. It converts kininogen into bradykinin, and now you all will understand that why did I call it the kinin system. Why did I call it the kinin system? Because what kinin have I formed in the end? Bradykinin. So if you don't want to learn this pathway, don't. There's no compulsion on learning everything. Itna you can learn what is the starting point and what is the last point. So who activated it? Factor 12. And what is the last point? That is bradykinin, because they will ask you what does bradykinin do. So please remember, bradykinin is very famous for 2 things which I want all of you to know very carefully. One is going to be vasodilation. Bradykinin causes vasodilation. You learn that, you don't learn that is a separate thing. But bradykinin causes pain. Because now all of you are going to make a table for me and tell me that what are all the things that I have taught you in this mediator thing which was causing pain. Do you remember which PG? I gave you one mnemonic also. There was one PG, that is PG. E, the way you make a sound, E causes pain, and even bradykinin. Now we've learned even bradykinin causes pain. So what are the pain producing things? The pain producing things are bradykinin and PGE2. So with that you will realize you have wrapped up your entire mediators, be it the cell or the plasma. In cell you've done preformed, newly synthesized, over plasma you've done all the Ks, complement, clotting, kinin. All of that is sorted. Obviously, you are ready for some MCQs now. It's an MCQ test which is going to have a lot of overloaded brain information for you, right? Gelatinase. Now, do you remember gelatinase was a lysosomal enzyme, and which particle or which granule was releasing it? Primary granule, specific granule, C particle, or azurophilic? What was your mnemonic? You can only help you, I can only help you with mnemonics. So there was something called C particle, and from the C we decided that we will make a G. So gelatinase comes from which particle? C particles. Can you tell me C particle was which granules? Primary, secondary, or tertiary? C particle, third alphabet. It was the tertiary granules. Very good. I'm sure all of you have answered it. Amazing. Let's move on to the next question for practice. Which is the most fibrogenic mediator? TNF alpha, TGF beta, cytokines, or prostaglandins? I hope you all remember. When we say TGF beta, बहुत fibrosis. So बहुत fibrosis is going to come down to TGF beta. If I would have asked you, that's the answer. But if I would have asked you which of the one is going to make you not make you hungry, no appetite. If no appetite would have been asked, you would have marked TNF alpha. Let's pick up another question before we go on to chronic inflammation, but a much needed question. CC includes which of the following chemokines? CC. So this was quite a tough table. Let me just highlight all these four. Can you tell me interleukin 8 comes under what? So I remember that mnemonic. CXC had interleukin 8. From the X we had made an 8 out of it, if you remember. Yes, okay. Now we come to fract alkene. Fract alkene was that weird one, that weird sounding structure, CX3C. Lymphotactin, L looks like a C, so that was just a C chemokine. So this means they have asked me CC. CC is going to be eotaxin. Eotaxin is for attracting which cell? EO, taxi, getting which cell in a taxi? Getting an eosinophil. So it is going to attract eosinophils. Well, yes, having said that, we finished all the mediators. Quite a dry but a very important part of inflammation is done. And now moving forward, we are left with two topics: chronic inflammation and wound healing.